few sentences about the International Group of Applied Preventive Medicine. Actually, we are in clinical research. We do support clinical labs. We're networking and clinical instructions, and I think discussing really.
Yeah, just
from this one, yeah. And so we do have academic educations running as well, all over the world. Actually, what we do is the first master course with the university. So we offer online master courses in preventive medicine. If you want to register, you will be welcome to do that. So my topic today, of course, is epigenetic hacking and longevity. And I was just referring to the fact that we would like to make it as simple as possible, but not really simpler. So I'm going to stay with the basics of longevity science. And I would like to refer to some practical applications in this regard. Biohacking epigenetics. What does it actually mean? I've just listened to your very interesting lecture with outstanding information. Thank you. But we believe, and please correct me if I'm wrong, everything is connected to this. What am I referring to? It is referring to energy. You can forget about senescence, you can forget about longevity if you don't have enough energy. So basically, our practical science, our clinical research is concentrating on mitochondria. And here we're coming up with some new facts about mitochondria, as we believe that mitochondria dominate your life and your quality of life. And if you want to know how old you are, biological age, you need to look into your mitochondria, nothing else. And this is very, very easy to do, I tell you. So I think we know that food is really resolved in the mitochondria, and most of the ATP produced is coming from the mitochondria. So energy, life energy, the currency of life is really closely related to the mitochondrial function. And here it is that this paper was published showing that mitochondrial dysfunction in cell senescence and aging is very closely related. So if you want to talk about senescence, you need to have a focus on the mitochondria and the function of mitochondria. And this is our message. Mitochondrial function determines length of life, and also, of course, the quality of life. The different sources that we know coming from a dysfunctional mitochondrial action are immense. You wouldn't know about any non-communicable disease not related to mitochondrial dysfunction. All of our non-communicable disease, be it diabetes, Alzheimer, whatever neurodegenerative disease, is closely related to mitochondrial function. And being an immunologist, I can tell you that everything about Th1 and Th2 or whatever you've been talking about is dominated by your mitochondrial function, because these are switching Th1 and Th2 functions. So I believe that we do have a message here to convey that mitochondria determine the amount of process of signaling, actually. And you see here the mitochondrial dysfunction is supposed to increase the loss, the apoptosis, and also, of course, ending up with inflammation. So all about COVID and the bad outcomes about COVID, they were related to mitochondrial dysfunction, actually, because we were not able to stop inflammation. We could have if we had some more things in mind than just applying these vaccines. So mitochondrial function, just to give you a short overview, of course it is ATP production, it is redox signaling, which is a strong key, very important key to life. It is nutrient synthesis, it is cell death, increasing, it is gene regulation. So what are you talking about? Gene length? This is dominated by your mitochondria. And if you want to talk about epigenetic impact, you have to talk about mitochondria because they are dominating your epigenetic impact. Everything that is closely related to your epigenetic impact is sitting in the mitochondria. So mitochondria do have a critical role in the immune function. I was just explaining that the switching of Th1 as Th2 is also critically related to the biogenesis of mitochondria. I will explain in my later slides. And this is about triggering non-communicable disease, also closely related to mitochondria, because, of course, if there's a deficiency of energy, the cell function drives you down. This is easy to understand. Here we have some more just papers. You might as well check PubMed, and you see so many of these publications coming up now. You see that this field is very, very broadened up by research, and so we thought to put this into clinical research now and to apply these theoretical understanding in your clinical applications. So what is the use of talking about experimental data but not using this for our clinical appliance? So I think we need to be based on our clinical understanding in order to approach our patients in a correct manner. The mitochondria are determining the inflammation, and cancer. Cancer is related to mitochondria. How many cells make it up without any mitochondria function? There are only two.
8%, red blood cell.
Right? And there's another one. Cancer cells. Easy, huh? They got without mitochondria, and once they have abolished mitochondrial function, they go ahead with their own way of building energy, which is glycolysis. So again, it's a very close relationship to all these things, you know, and the mitochondrial function is very closely related to neurodegenerative disease, cardiovascular disease, immune deficiency, cancer cells, mitochondria. So everything that you've been discussing about, it's about mitochondrial function. Metabolic disease, if you increase your body fat, it's a dysfunctional area of your mitochondria, actually. So if we look closer, I think everybody knows this. Mitochondria do have two membranes, the inner and the outer membrane. But did you know about this here? Mitochondrial DNA, what are they supposed to do? Of course, they are controlling the biochemistry of mitochondria, the respiration, but I will tell you some more information about this mitochondrial DNA and how it really looks like, what it's working. We do have mitochondrial DNA. So they have their own genome, of course. They are double-stranded but not methylized. It is that they have codes for 37 genes. They are not able to repair themselves. Important. They are not able to repair themselves. So once you hit, they have oxidative stress. If you put your mitochondria under oxidative stress, they are not repairable. They are very susceptible to loss. And of course, they are in every mitochondria. How many mitochondria do we usually have in a cell? Just make a guess. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10? No, thousands. Usually thousands, depending on the organ, depending on the function of the mitochondria, actually. So this is maybe a new thing for you. that its latest research showing you, telling you the importance of mitochondrial DNA. As I was bringing out that there are 37 genes and base pairs in the mitochondrial DNA. They're regulating the ATP production. Okay, that's sure. But did you know that it seems that they influence the process of DNA nucleus as well? Yes, they are determining what your reading of your DNA is. Because once they leave the mitochondria, they're implemented in your nucleus DNA. And there they start the reading of your DNA. And so maybe these mitochondrial DNA this is our latest finding actually, they determine what kind of metabolic onset you have. So mitochondria and mitogenesis, what we call, is the homeostasis of mitochondrial production. And this is now they are living organs. Mitochondria are not static, they're very dynamic. What they do is they love to fuse and they love to have fissions. So they're moving and they are replicating themselves. It's not a stable, it's a dynamic process. They are in a dynamic process. And because of this, they are able to exchange information. They are able to repair. They are able to repair the dysfunctions. So it's very critical that we, let me see if I can start that one. You see that they're moving? Look up here. This mitochondria under fission and fusion. And this kind of dynamics, they have an impact on your immune system. Because the phase that they are just undergoing determines you if you're Th1 or Th2. So this is very important that mitochondria actually determine your immune system. So just coming to the point that I'm not only talking about mitochondria, I could explain a little bit more. It is fascinating actually. This chapter is so very interesting. But my topic today is how to have an impact on the mitochondrial function. And as I was explaining today, I would like to do it as simple as possible. So one thing is not using medication, because this is a fancy thing. You can go and have some drugs supporting the mitochondria. We like to talk about nutrition. Nutrition is available for everybody, and you can adjust your mitochondrial function if you select yourself the right nutrition. Then I would like to refer to additional concepts, and then very special for you only about the individually tailored supplements for mitochondrial health, because you would like to know how to prescribe these things. So what is the mito diet actually, mito nutrition? Well, it's nothing more than just low carb. It is actually more or less fasting, could be intermittent fasting or whatever. It is at least reducing your calories. And it is glucose restriction, because this really, and this is science, we know that, really delays senescence and promotes proliferation of AMPK insert signaling. So very easy. Keto diet is a ketogenic diet that provides approximately 75% of total energy intakes from fat, 70% from protein, and only 5% from carbohydrates. An average person would need about 2,000 calories, let's say, so per day. This is why he is allowed for a maximum of 50 grams of carbohydrates on this diet. Ketones are produced in fasting, and ketone bodies are composed of three metabolites, namely beta-hydroxybutyrate, pseudoacetate, and acetone. These ketone bodies are very friendly to mitochondria because they support mitochondria respiration. These ketones are in charge of supporting the mitochondrial function. They support the metabolism of the various functions of the mitochondria, including the signal transduction and the epigenome regulation. So we've been talking about epigenome biohacking, right? Have you heard about regulation of mitochondrial protection? Ketogenic diet will be one step, which is very easy to do, about carbohydrate restriction. So there are so many things to explain. Because of lack of time, I'm not going to get into these all. The synthesis of ketone bodies is coming from the mitochondria and the degradation of ketone bodies as well. So ketones, they love mitochondria. And we produce ketones, we will have this very significant signaling in our body, which is called the AMPK signaling. It's a cellular signaling and a mitochondrial signaling, actually. AMPK acts as a signal to control mitochondrial biogenesis, and AMPK regulates at least 3 essential aspects of mitochondrial homeostasis. This is the biogenesis, the fusion, and the mitophagy. I was just showing you the picture that the slide that the mitochondria are just in diversion. They are moving, they are changing, they are under fission and fusion. And this is controlled by AMPK. And of course, we know that AMPK inhibits mTOR and activates autophagy in the mitochondria during carbohydrate restriction by inhibition of mTOR1. So this is, of course, well known to you, AMPK and mTOR. So we like to support the AMPK signaling, and this is easily done if you are on glucose restriction. The length, okay, that's different. You might do this in an intermittent fasting or for a longer time. Dr. Omo was just explaining 5 days at least. That's helpful. Anyway, the signaling is off and on. You can do it, and by this, every now and then, just do your glucose fasting, and you will increase your senescence without any other help, of course. AMPK activators. So we're coming on to the point that fasting is one thing, but the other thing is, do we have any other remedies at hand to also increase the AMPK signaling? Yes, we do have. This is just an overlooking picture about AMPK signaling, and you see that AMPK is controlling ROS in ATP production in the mitochondria. Please have a look up there on the oxygen and glucose deprivation. It is hampering. It is hampering. So we would need to see that glucose restriction is very helpful in supporting AMPK. So nutrition number 1, but then we do have additional supporting general concepts of mitohomeostasis, and I'm going to have some minutes on this one now. We've had fasting, then we have the external ketone bodies that we can use, and then I will explain some more things. So the ketone bodies, if you cannot really go to fasting, you can use these ketone bodies, external ones. You can even check your ketone levels with the help of these machines. Do the keto inhalation test, which is very simple to do. And then the advantages of ketones, I've explained that before. They are supporting the whole respiration and the biogenesis of mitochondria. Mitochondrial function is under control of these ketones. It is a very simple thing to produce ketones while fasting, but there's a catch. What is this catch like? If you reduce your carbohydrates in your diets, you will reduce your very, very important activators in your gut, because they are also involved in this whole chapter. So microbiome is having a strong impact on your ketogenesis. And you need to see that there's a crosstalk between the microbiome and also the mitochondria in your whole body. How do you improve the reading from the microbiome and produce more ketonic substances in your body? Well, you need to have insoluble fiber and fermentable fibers in your diets as well. Because dietary fibers, they include gut-produced butyrate, insoluble dietary fibers with specific chemical structures that are helpful to produce these ketonic bodies. The butyrate produced during fermentation supports gut barrier function through the stimulation of glucose production in tight junction, and thereby reduces inflammation in the gut. And the butyrate also regulates inflammatory pathways through heat protein coupled receptors in enterocytes and through inhibition of histone deacetylase in the macrophage. GPCR induces secretion of hormones, glucagon-like peptides, and all these hormones that are driving senescence and driving your signaling system. So this is very important. If you are under glucose restrictions, you better use also fermentable fibers, including pectins, beta-glucans, guar, inulin, or oligofructose. These things are helpful to support your gut flora because they are the main players in your gut, not only just reducing your glucose in your diets. So the question is, how do you produce resistant starch?
Store away your rice, eat whole rice.
Oh, you can eat rice. There's a trick to it.
You know, whole rice. Your recycled rice.
Recycled rice, yeah. You cook it,
You store it,
and then you eat it again. That's it. It changes the chains of the rice and the starch, and this is good for the bacteria because they cannot dissolve these chains anymore. So good, yeah. Fermentable and non-fermentable fibers, they support your ketone production in the gut. There are some things that you can just use for mixed pickles, and they're also supporting, but when you look around and you just realize the smell, you know the body next to you is producing some more bloating because it's using fermentable carbohydrates. Yes, I would like to share this message with you that we have two ways using the diets and also using the ketogenic exogenes, but also we would like to look into individually helping the mitochondria by supplements. How can we just supply the supplements in a way that they are individually tailored? Because I tell you, not everybody has the same need in the supplements. You have to be very individually precise about what we do. This is the new word called precision medicine, actually. This has been already referred to by Dr. Homer. Resveratrol is very helpful. Quercetin is very helpful, and alpha-lipoic acid, which you can use as an infusion as well, is very helpful. And then we do have, of course, another thing coming up in Europe. We're using spermidine infusions. Spermidine infusions, they can activate the cellular recycling process of autophagy. Very helpful, very easy to do. It can even slow down the aging process. And it was also inhibiting SARS-CoV-2 autophagy in the infected cells. So this research at the University of Berlin was very helpful to bring this substance out, and it's available in Germany as an infusion concept. Spermidine infusions, they are GPMPT tested, so this is a very easy way of driving your senescence. It's registered, you can get it from the drugstore, pharmacies actually. But I was just referring to the fact that we would like to, be individual oriented in our treatment. We would like to use some hallmarks of aging, and here we have the mitochondrial dysfunction as a hallmark, and we would like to check the mitochondrial function. I will just give you one test. We do have more tests available, but this is an easy test that you can apply in the Philippines as well, because sending to our lab won't be that difficult. This is a urine test. This is a urine test, a urine analysis about the metabolome, and it covers all biochemical pathways of the mitochondria actually. It is a metabolic cycle analysis of the organic acids. Here we just have the reading that all these different acids that are analyzed in this urine, they're giving us information which kind of blockages you have in your pathways of the mitochondria. So easy test to sample, and you get the result from your patients. So here's just an overview of different acids that we just been able to differentiate, and this is of course related, of course, to the instructions that you can use. So here, for instance, if you look at the citric acid, it calls for a magnesium and vitamin B12 deficiency. This is just an example. You read the succinic acid, so it tells you have a vitamin B3 deficiency. So this is very helpful to go along your treatment with individualized, tailored aspects. And I'm just going to give you a very short overview of the case report, just to explain how this really works. It's a 9-year-old schoolboy. Mother is worried about lack of learning performance and lack of concentration. Intelligence test results fair. No obvious deficiency, but behavioral overactivity. Psychiatrist says attention deficiency syndrome. So what we did actually, in this case, we applied this urine test. Very simple. Because it was a boy, we needed to take blood from this guy, and this was the example that we had, patient, and then we came out with this reading. He had high levels of adipase and ethylmyelinate. These were actually standing for L-carnitine and vitamin B2 deficiencies in this specific case. So what we applied was, of course, L-carnitine in this dosage, B2, and of course B1, magnesium and manganese, and that helped him to get a stable outcome. So my home take-home message today is epigenetic-biohacker should have to focus on functional mitochondrial function. I do have more tests at hand to be precise about mitochondrial function, but they take more. You need to take blood from them, but these are available as well. But I was just explaining that the urine test works very nice as well. Mitochondrial function depends on the proper diet, or we should stop eating empty calories, and the mitogenesis is mainly controlled by two subcellular antagonists like AMPK and mTOR. We're just discussing how to improve the balancing. Fasting, carb restriction, and essential amino acid support in favor of EMPK signaling and longevity is closely related to healthy mitochondrial function. Thank you very much for your attention. It's time to break.
Okay, time for our